The mucilage covering, remember one thing, that a cell wall, outside the cell wall, a bacterium will either have a mucilage covering, a slippery covering, or otherwise there will be a hardened structure called as capsule. So one of these structure is always present in the bacterium. A gram-negative bacteria, which are basically pathogenic bacteria, they have a layer of mucilage made up of polysaccharide and lipid outside the cell wall, and it could become hardened with the nitrogen containing compounds to form a capsule for protection, which provides mechanical as well as the physiological protection. Now the next component is the cell wall. The cell wall contains the variable amount of peptidoglycan and lipid, as we have covered. Accordingly, they are either called as gram-negative or gram-positive bacteria. The cytoplasm contains the glycogen. Cytoplasm contains the food droplets, which are fat droplets. These are the food, reserve food material. Cytoplasm also carries ribosome. They can be single ribosomes scattered, or they could be arranged in the chain-like manner on the mRNA, which is called as the 70S type of ribosome. Remember, prokaryotic ribosome is 70S always. RNA is always also present, and the photosynthetic species of bacteria will carry bacteriochlorophyll in the thylakoids. Remember, chloroplast is absent, plastids are absent, but very simple membrane-like structures carrying pigment, which is called as bacteriochlorophyll, is present if it is a photosynthetic bacteria, and such layers or lamellae are called as the thylakoids. Now, nucleoid is the double-stranded naked DNA present inside of the nucleus. Plasmid, what is plasmid? Very important component. It is the additional ring of the DNA, double-stranded DNA, which replicates independently and provides special characteristics to the bacteria. A plasmid, and remember, special characteristic means nitrogen fixing ability, sex duction, or what we call as the sex pili or sex fertility ability, and then they even provide resistance against the antibiotics. So plasmid is not necessary for survival, but it provides special features. A plasmid which get temporarily incorporated into the nucleoid is called as episome. Sometimes the plasmid attaches to the nucleoid. If it attaches it there temporarily, we call it as episome. Cell membrane is made up of the phospholipids, proteins and sugars. It is selectively permeable and has following modification. So cell membrane will carry flagella made up of flagellin, which is a protein. Remember, there is no tubulin present here. Tubulin structure is found in the eukaryotic flagella. Flagellin is the protein found in the prokaryotic flagella. It is 4 to 5 micrometer in length, and it is required for locomotion. Pili is made up of a protein called as pilin, which is 2 micrometer in length and is found in the donor bacteria. So those bacteria which are able to form pili will act like a male or donor, and those which do not develop them are called as the female or the receiver. And with the help of this pili, genetic material is transferred into the female acting bacteria by the process called as conjugation, a topic which is basically not in your syllabus now. The mesosomes are the finger-like projections which develop towards the inner side of the plasma membrane, which goes into the cytoplasm and help in cell division, help in providing more surface area for the respiratory enzyme, and it also help in the proper nucleoid or the DNA replication. There are respiratory enzymes all over the inner surface of the cell membrane. Now, reproduction. Monera, the organism which belong to Monera, they perform binary fission. It is the most common method of multiplication in bacteria under the favorable condition. So you can see here, mesosome is attached with the nucleoid, and when the copy of the nucleoid is formed, in the same time the copy of mesosome formation also takes place. And finally, from this common point, the membrane starts moving insides, and slowly this will divide into 2 equal cells. So this is how cell division or binary fission takes place. Now, there is another method of reproduction that involves sporulation or spore formation. Spores can be endospore, oidea, conidia, gonidia. Cysts are the most common type of spores found in them, which is called a sporulation process. And then I told you there is a very different type of recombination or genetic recombination possible in bacteria, which is called Aspera Sexuality. It includes conjugation. It was first discovered in the E. coli by Liederberg and Totem, and such bacteria show dimorphism. One bacteria is called as positive or male or donor, and the other one is called as the F negative or female or recipient. Remember, those which will have the F factor in their plasmid will act like a F positive one. Those which lack is called as the F negative one, and those which carry this F factor will develop the sex pilus. So a pilus like structure will develop, which will cause the genetic material to go from one cell to the next cell, and this is how no number increase takes place, only the genetic material is mixed. There is another type of conjugation which occurs in the HFR. HFR bacteria are those which have a higher frequency of recombination. Now the next thing is the another type of parasexuality called as transformation, which comes in your 12th class syllabus. It was first discovered in Diplococcus pneumonia by Griffith in 1928, and in this process a bacterium picks up the genes from the dead and decaying bacteria in the surrounding, which comes in details in the 12th class, and transduction also comes in the 12th class. It was first discovered in the Salmonella typhimurium by Zinder and Liederberg, and in this process a bacteriophages are those viruses which attack bacteria, so their hosts are bacteria. So in this process a virus is going to transfer the genes from one bacterium strain to the other bacterium strain. Now respiration. Bacteria respire either aerobically or anaerobically, and they can be obligate. Obligate means 100% aerobic. They cannot live without oxygen. And then there are some which are basically aerobes, but they can turn into anaerobic situation if there is requirement. So they can survive somehow, sometime in the anaerobic condition. So accordingly they are called as facultative anaerobes. Facultative means sometimes. Now second type is the anaerobic bacteria, which only survive in the absence of oxygen, so they are obligatory anaerobes. And those which are anaerobes but they can somehow survive in the aerobic situation are called as facultative aerobes. Now nutrition. Bacteria are either autotrophic, including photosynthetic, because plus remember there is no oxygen liberation in the bacterial photosynthesis, so it is anoxygenic photosynthesis, or otherwise they perform chemo-autotrophic nutrition. If they are heterotrophic, they are either saprophyte or symbionts like rhizobium, or otherwise they act like a